Okay, so hello everyone. Thanks very much for inviting me. I'm really sorry for not being there with you. So the ultimate goal of my talk is to somehow show that mechanistic modeling still has value in a scientific background where more and more we talk about artificial intelligence. So for that, I will present you some of our recent results. I will start with our work on meningioma. So as you probably know, meningioma are intracranial tumors, not originating from the brain, but just outside the brain. They are slowly evolving, but unfortunately, they could be very badly located, for instance, just behind the optical nerves or structures like that. And the standard of care is a curative surgery after a variable follow-up, because if the tumor is not growing too much, maybe there is no clinical need to resect it. So clinicians tend to wait for having an idea of the evolution of the tumor before removing it. So the clinical challenge is to schedule the checkups and the monitoring, because you don't know exactly how many time you have between a significant growth, and predicting risk of evolution is very key to improving the standard of care of the disease. So this is why we started a project with the Bordeaux hospital, whose goal was to try to see if with mechanistic modeling we can build something that could add insight or information on the evolution for the clinician. So this is a joint work with many nice people, starting from Thierry Collin, Annabelle Collin, mathematicians, some postdocs, Cédric Coppol and Vivien Pianet, and just starting PhD student Virginie Montalibé, and some clinicians from the Bordeaux hospital, Hugues Loiseau and Julien Engelhardt. So typically the challenge we have from a clinical point of view is the following one. You have a patient like the one here. You have a baseline exam where you can identify immediately the small tumor here on the right part of the brain, and this tumor is growing, and you want to predict the evolution of this tumor to be able to know if it will be interesting to remove it for the patient. So given the two first exams, are we able to predict the evolution, for instance, of the third one? So this started with a little less than 50 patients from Bordeaux, and for each patient we have various information, but at least we had 3 exams per patient showing delineated tumors. So from that we could compute volume, we could just analyze the texture of the tumor, we could maybe have an idea of the location of the tumor, and also we had some clinical information like age or sex, and most of these patients are female patients, because this is a pathology that is more frequent for women than men. So if we were to build such an algorithm for predicting the growth of the tumor, there are different strategies that one could think of. The first one is to use mechanistic modeling. So basically, it means that you want to turn the disease mechanism into equation and start to analyze this equation to build the prediction. Or you could turn to more fancy techniques like machine learning techniques. We could analyze the texture through radiomics, for instance, or we could even use deep learning techniques to build neural networks to try to predict the evolution of the disease. But here, the problem was that, as most of the time for clinical cancer problems, you have small cohorts, so it's difficult to build very precise learning algorithm when you have only a few patients in your data set. Also, the tumor were quite homogeneous, so probably that the texture analysis would not have been so significant if there is no variability of texture among patients. And you definitely want to use the longitudinal follow-up. So most of the time, maybe at least for radiomics, it's not so useful. The radiomics are not so efficient to use this longitudinal information. So we turn to mechanistic modeling, and we try to build a mathematical model of the natural growth of meningioma, because as I said, there is no treatment for this patient, which makes it a very interesting problem to work on. So you start from medical knowledge to build your mathematical model. In this case, it was quite simple. The meningioma is growing out of the arachnoid, which is just a small structure stuck to the skull. The evolution is slow and completely 3D because it's growing in the just outside the brain, and the tumor is quite homogeneous. And as always, you have to do some trade-offs, some modeling trade-offs, because you want to have an accurate description of the disease and its evolution. So for that, you need to have a mathematical model that is complex enough that can produce reproduce the behavior you want to observe. But also you want to calibrate this model on patient data to be able to do some prediction. So you have to find a way to recover all the parameters that are included in your model. And unfortunately, for this kind of clinical model, there is no way to directly measure this parameter or even to relate these parameters to biological quantities you could find in the literature, for instance. So you have to make this trade-off, and in this case, as we have only 3 exams, it's quite simple. And the kind of model you can use is this one. So this is a model that is somehow derived from the Gond-Perse model. We describe the evolution of cancer cell densities, denoted by T. And this quantity follows an advection equation. So this kind of model were made familiar by Luigi Preziosi and Davide Ambrosi. And in this model, we have a growth rate, or in this case, it's more something that we could relate to vascularization, here denoted by M, and we say that the vascularization is decreasing over time because the tumor is consuming nutrients and so. So obviously this is completely simplistic, but if we want to fit this kind of model to patient data, we have to accept that the model will be very, very simple. And outside the tumor, we consider there is healthy tissue, which is just undergoing the pressure created by the cellular division of the tumor cells. And finally, we close the system by assuming that the velocity is following a Darcy's law. So we could use a different closure. For instance, we try to play with Stokes closure or even viscoelastic closure. But if you do that, you increase the number of parameters in the system and you make it more difficult to fit the model to patient data. So in the end, with this simple model, we have 2 parameters to recover, and these parameters are completely patient-specific. You have no way to measure them outside from the data you have. So the real problem is the following one: given this simple model, how do you calibrate this model, or how do you recover reasonable values of the parameter that allow you to reproduce the observation you have from the clinics? So in our case, the observation are just the MRI exams, so we want to use that. And in the end, we try to find strategies to recover the parameter. So for that, there are many methods available. You can think of gradient-based method, Monte Carlo method, or many other methods. But the key thing is that almost all these methods require a large number of simulation. And if you remember, the model is supposed to be completely 3D. So you have a PDE in 3D to solve around the tumor. It could be very expensive. So if you want to calibrate this model using such method, it could be very time consuming and also not realistic for clinical use. So model personalization is that you have model, you have data. So in our case, data is the tumor fronts at exam dates as delineated by the radiologist or the neurosurgeons. You have simulation, given a set of parameters, you are able to simulate your model, and then you compare the simulation and the observation, and you try to minimize the difference between the two. And here, in our case, there is a small miracle, because the 3D model can be reduced to an ODE model by integrating over the tumor volume. And this means that you get a reduced order model with the same parameter as the PDE model, but you can use this reduced model that describes the evolution of the tumor volume to have a prior on the value of the parameter you could put in the 3D model. So obviously, this is not completely sufficient, because if you do model calibration per patient, you lose all the information you could have from considering the whole population of patients. Initially, we had only 50 patients, but now we have, I think, more than 400 patients. So definitely, you want to consider the whole cohort. And for doing that, as we now have a reduced order model, which is a PDE, we can use popular techniques like mixed effect models. And in mixed effect model, basically what you do is that you decompose each parameter in two parts. One part is shared by all the patients. So this is where you put the information you get from the population information, and a small individual fluctuation around this average that describe the patient-specific value of the parameters. And to recover that, we use a popular algorithm that is the SRM algorithm that is implemented in monoliths. And from that, using the volume of the patients, you can get a meaningful prior for the PD model. Obviously, as always, there are a large source of uncertainties in your problem. For instance, if you remember, the observation we have are tumor fronts from MRIs. We only consider tumor front because, as I said, the tumor is quite homogeneous. But this segmentation could be noisy. They could have some uncertainty. And if you take two different neurosurgeons or maybe two different radiologists and you ask them for segmenting, probably that there would be some small difference between the segmentation. So you definitely don't want your algorithm, your predicting algorithm, to be sensitive to this kind of uncertainty. And obviously, your model is not perfect. The calibration is not perfect as well. So there is a huge sources of uncertainties. And what you want, if you want to build an algorithm that is helping clinicians to follow the disease, you want to control these uncertainties and the effect on the response of the tool you give them. So first, from this most simple model, from the calibration technique, we start by using that to validate the model. So in this step, what you want to do is to check to be sure that the model is able to reproduce all the observed behavior you have in your data set. So this is somehow the validation of the descriptive aspect of the model. And on the right part of the slide, you have for different patients, you have the black dots that are describing the observed values, the observed tumor volume made by neurosurgeons, and the red line is the fit of the model on all these examples. So as you can see, for most patients, we are quite good. So the results are satisfactory. It means that at least your model is able to reproduce the data you have. Also, you can observe that the growth of the meningioma are not the same. Some patients, there is a small decrease at the end of the observed period. For some patients, it looks like a linear evolution. For some patients, it's more exponential. So there is some kind of variability among patients. And so it means that even if the model is simplistic, a simpler model probably would have not worked for this patient. And once you have validated the descriptive aspect of the model, now the real question is, can we use this model to do some kind of prediction? So here, what we use is that with the output of the mixed effect approach, we have some priors on the parameter. That means that even if we have a new patient with only 2 exams, we can use this prior to get some estimation of the evolution of the disease that we can correct with the 2 first exams. So this is exactly what we are doing here. And this allows us to do some kind of prediction for patients given only 2 exams, and the results is shown on the right figure of the slide. So you can see that each point is a patient. On the x-axis, you have the measured volume by neurosurgeon at the 3rd exam, and on the y-axis, you have a predicted volume. So basically, if you are on the x equal y line, you are perfect, and you can see that here the prediction is not so bad. And then, as I said, we have a reduced order model. So we get from the PD, we get an ODE model, we calibrate the ODE model, so we get parameters from the ODE model, but these parameters are the same for the 3D model, and we can use this parameter to start as a prior for the 3D model. So this is exactly what we have done here, to have some 3D simulation of the evolution of the disease after calibration. So here you have different patients. In blue, you have the segmentation that was done by neurosurgeon, and in red, you have the simulation produced by the model after calibration. And you see that we are not so bad. And on the right part of the slide, you have the dice coefficient for some patient, and you can see that we are very good, at least on this patient here. So we have a good prediction of the volume, we have a good prediction of the shape and location, but now what could be what could we do with this work? So first, we could improve data assimilation and model personalization, especially because here there was a miracle, the fact that we could reduce the model to an ODE. So this is something we are interested in. There are some works to translate at least to adapt a mixed effect model for PDs, which is a quite complicated topic. So Emmanuel Grenier did something in Lyon about that. And also we are working on sequential methods. For instance, we did some interesting work on for brain metastasis, without treatment as well. A key thing of the work was with the reduced order model, as we said. So we also work at trying to find some ways to compute the 3D simulation in a more efficient way. And finally, obviously, if you want to use that at the clinical scale, we have to do a larger and possibly multicentric study for validation. So this is ongoing. This is currently happening in Bordeaux and hopefully with other clinical centers. So now we are essentially each time there is a new patient with meningioma coming to Bordeaux, we plan to use this data to improve the model. And what we want to do as well is we want to combine the non-radiological features. For instance, I said we have the age of the patient, we have some clinical features like hormonal treatments or things like that, and we want to combine that using the model. So one way would be to combine the mechanistic modeling to get the parameters that are specific to each patient, and to combine that with the other features through, for instance, a machine learning algorithm, because now we have a significant data set of more than 400 patients. And as I said, also what was very important in this work was to have the tumor front over the exam, and this is not something you can ask neurosurgeons or radiologists to do in routine. So you have to find a way to automate all the segmentation work. So this is why in parallel, we are working on algorithm to automatically delineate meningioma. And all this, it would be the goal of the thèse of the PhD of Virginie Montalibé. And just now, I want to show you a different example for another pathology, which is a non-small cell lung carcinoma, which is a quite frequent lung cancer with a very bad prognosis. And there is a huge number of therapy that could be given to a patient depending on the stage of the disease, the history of the patient. And so the clinical tree, the decision tree for clinicians is very complicated, because you have, for instance, chemotherapy, surgery, radiotherapy. You could use targeted therapies like TKI, immunotherapy, many things to do. And in fact, the clinical problem is, in this case, would be to what is the best treatment for the patient given his history. So one thing we try to do is to offer some kind of early evaluation of therapy efficacy to clinicians. And we had this project with Institut Bergonier, where we try to improve the evaluation of the efficacy of tyrosine kinase inhibitors for EGFR mutated NSCLC, which is only a small part of the lung cancer case. And unfortunately, for this patient, the treatment is only temporarily working, and after a while, there is almost always a relapse of the cancer. And the question was, can we predict the time to relapse for this patient? So here you have a typical NSCLC for this patient. You can see we have 3 times here for a patient, and the tumor is under treatment, so it is shrinking a bit. And in fact, what we want to do is we want to predict whether the relapse will happen before or after 30 months. And for that, as before, we have built a PDA model for response to ITK. In this case, it was not completely necessary. We could use directly an ODE model, but what we wanted to use is we wanted to exploit the radiological images maybe more than it was currently done. So for that, it turns out that maybe it was not so clear from the previous images, but the tumor is less homogeneous than that, for instance, for meningioma. And there seems to exist some kind of influence of this heterogeneity of the survival of the patient. This is why we start studying the images maybe with more depth. So we postulate that there is 2 kinds of cancer cells inside the tumor. Some are proliferative, some are quiescent, and our assumption is that essentially we can distinguish between the 2 by looking at the voxel intensity within the tumor. So that means we took intensity, high intensity within the tumor. We said that it is proliferative cells, and lower intensity, it is quiescent cells. So from baseline CT, we got this initial distribution. And it looks like if you take the integral of the proportion of proliferative cells within the tumor, it looks like it could be a meaningful biomarker of the evolution of the disease. But unfortunately, if you only compute this biomarker at the exam dates, for instance, if you compute at T0, T1, T2, this marker is very sensible. That means that if you change a bit the exam dates, or sometimes the scheduling is very variable. You have 2 first exams are separated from many months, while the third one is very close to the second one. It's difficult to compute reliably this biomarker, and also essentially, if you compute an integral, it means that you are doing a linear interpolation. So our goal was to see that if we have a model for the evolution of the tumor between this point, we can improve this biomarker. So this is exactly what we have done. We have built this model with 2 different kinds of cancer cells. We have fitted the model on patient data, By the way, you can see that the observation of the tumor may be quite different between the patients. And from this calibrated model, we got the parameters, and we computed the integral of the proliferative part of the tumor between the exams. But this time, the model gives us an interpolation of the evolution of the disease between the exam dates. So the evaluation of the integral you have is more accurate because you have some kind of biological constraint of the evolution of the disease between the exam dates. So this is what we have done. We have computed these biomarkers, and it turns out that this biomarker is able to improve a bit the classification of patients. If you remember, we want to say whether a patient will relapse before or after 30 months. And if we use our model to distinguish the patient, we can see that we have quite good classification results. And we are able to separate the patient in two groups depending on the value of this parameter. So compared to a purely imaging-based approach where you just compute the evolution, the proliferative part of the tumor and build a marker from that, our method seems to be more robust. But as always, obviously, this would have to be validated on a much larger scale. So this is the goal of a project that is funded by MSD, more or less, which is a big pharma. And as always, we want to see if it can be deployed in clinics. So as I said before, you have to automate the work of segmentation, because if you don't do that, you will never have enough information to build a model or to build something interesting for clinicians. This is why we have developed deep learning techniques for that. There is a huge variability of lesion size, so you have to be careful about having an algorithm that is able to segment lower stage where you have very small nodules that are very easy to distinguish on the radiological image, from larger stage or grades where the disease is almost completely invaded the parenchyma. So in this case, the talking about maybe segmenting a disease is maybe more difficult completely. So we have done something like for this problem. So we started with a classical network for that unit. We use a cascading approach to start with from the whole image, then to refine using a patch approach inside the targeted lesion. So I won't spend much time on that, but what I think is important is that you cannot develop model at the clinical scale if you don't consider such challenges, because without data, you won't be able to prove that your model is right. So this is a pipeline that you have to work. You have to work on each part of this pipeline if you want to build some kind of interesting algorithm for patients. So maybe I'm a bit late, but we did. Sometimes it turns out you do not have a follow-up for patients, and if you want to answer meaningful clinical question, you have to build something. So this part, this example was just to show you that even when you have only one exam for a patient, you can do something, and what you have to do is. In this case, it is using machine learning. So if you have one exam, you cannot really build a model for describing the disease. So you have to rely on machine learning techniques. So in this case, this is something we have done for the same pathology, but for the response to immunotherapy. This is a project that was done with Tenon Hospital and Sophia Genetics for some kind of for immunotherapy for late-stage disease. And here we had promising results on 60 patients from the Tenon Hospital, and we are still working on that. But what is very important to consider is this whole pipeline where you could also insert the mathematical model. because if you want to give answers to clinicians, you have to work on it in each of these parts. So you have to understand what are the uncertainties you have during the acquisition of the images. What is the variability of the segmentation you use to calibrate your model or to build your machine learning algorithm? Then there is some kind of almost some kind of processing involved. So, for instance, for radiomics, you have a process to go from the image to the feature, the quantitative features. There is extraction of features that you have to control if there is some uncertainty in that. And then you have a large number of features. There is a process of feature selection. So you have to understand all the features you are selecting contribute to the final algorithm. Are they correlated? Are some of them more interpretable than others? And then you have your algorithm to validate. So if it is a mechanistic modeling, you have also the to calibrate the model, which could be tricky. And you have to combine all these sources of uncertainties to build an algorithm that is interesting for clinicians. So it could be very difficult. And also, obviously, you want to have an algorithm that is reproducible. So if it works in Bordeaux, it should work in Lyon or in Paris. So you have to work on that as well. So this is one part of our work. Is also to, maybe it's not the most I don't know, the most exciting part, but this is something that you have to do, is to study the influence of the different sources of variability on features classification mathematical model. So we did something on each of these parts: acquisition, pre-processing, as I said, segmentation, feature extraction, and also now we are working on techniques to do some kind of harmonization when you have multicentric studies. What are the large perspectives? So the main challenges in clinical oncology is that most of the time you have small data sets, because you are working typically on some very specific pathology. Sometimes it's difficult to have hundreds of patients for this pathology, and also there's obviously some privacy issues from dealing with patient data. Sometimes these data sets are highly imbalanced. It means, for instance, that you could have a pathology that is very rare. You want to predict if the pathology, if you have this pathology for patient, but then you have a data set when most of the patients are not sick with this pathology. Labeling is very expensive because you need to have some medical experience, some medical skill for that. And it's not something that is easy to find, because clinicians, they do not have the time to do that. So you have to work on some technique to maybe to rely less on labeling. So this is why we are working on methods like semi-supervision or auto-supervision that allow you to build classification algorithm with less labeled data. As I said, each model is more or less specific to a given pathology, which is very challenging. It means that if you change from, I don't know, a brain cancer to a liver cancer, the model will be different. Still, there are some similarities between the model, but the model is different. So it means that you have to start almost from scratch when you target a new clinical problem. And also, it means that the calibration technique could be different from the two approaches. Learning or data simulation obviously is a key and also very challenging. So as I said, we use some techniques like mixed effect model that are difficult to extend to PDs. When you have PDs, you have to find a way to have priors on the parameters you want to estimate. So one good way to have this prior would be to use, for instance, machine learning. If you have enough patients, you can identify some priors from data, which is something we are working on. If you want to be able to reproduce evolution over time of the disease, you have to resort to longitudinal follow-ups. Most of the time, it also limits the the pathology you can work on, because when there is a serious concern, the clinician won't wait for an answer from mathematician. So, for instance, if you work on highly aggressive cancer types, maybe it's not so interesting to do some mechanistic modeling for that, because it won't be used in practice. And you have to work on understanding or controlling uncertainties or robustness. And if you want to have an algorithm used by clinician, you have also to work a bit on interpretability. And I think that's it. I want to thank you for your attention. I'm very sorry for not being there with you. And yeah, I'm happy to answer any question you would have. Thank you.
Okay, maybe I can start. Yes, Olivier, so thank you very much for this nice presentation. I was wondering, how is the geometry of your solution dependent on the environment, actually? Why is the solution evolving in this specific shape? Is it because you prescribe like kind of this vascular network around, or the geometry of the brain, and how specific is it to patient then?
Yeah, for meningioma, it's quite specific because the tumor is growing close to a very hard structure that is not really invading. So there is like a wall on one side of the tumor. So this wall makes the evolution is not is depending on the location. And also for meningioma, we are interested in predicting whether dangerous part of the brain would be pushed by the tumor. So it means that you want to predict the location. For instance, if the meningioma is close to an optical nerve, you want to be able to tell that to clinicians. And so if the tumor is surrounded by smooth tissue, like in the lung, probably there is not much influence of the geometry, except when you are on the boundary of the lung. But for the brain, you have to consider the geometry. This is why probably you cannot really use simple model or that do not consider the spatial aspect of the growth.
Okay. And when you said that you need specific models for different kind of tumors, then it's because of this, or it's also because you have some other mechanisms that are at play? Like
there are definitely different mechanisms. Because, for instance, there is a huge difference between metastasis, when you have cancer cells from outside the organ growing in the organ, so you have quite homogeneous tumor, so you can, and most of the time the boundary between tumor and healthy tissue is very easy to see. On the contrary, if you study primary tumors, like as I showed, like this kind of lung tumors, or for instance glioblastoma or glioma, the cancer cells are within their own organs, the organ in which they live without turning to a rogue state. So there is some kind of invasion that you cannot really see on imaging. For instance, it's well known that for glioblastoma, there are cancer cells invading the brain. You cannot really see these cells on MRIs. So there is something happening under your control. So it means that you have different shapes, but the shapes are less regular when you consider late stage primary tumors. So, and also the organ, their own specificity. As I said, if you have meningioma, you have hard part around the tumor. If you have a tumor growing in the rectum or in the galactophore channel, you have different geometrical constraints. So yeah, obviously each cancer is different. Even there is obviously some similarities between solid tumor. So it means that you can recycle the modeling you have done on one part with to another one.
Okay, thank you. I think there is a question.
Yeah, you have a question in two. For the classification you showed, the group are of very different size, or
Yeah. This is just preliminary, and this is. I think this is an example of what is why it is difficult to work in clinical oncology, because here you are trying to predict the relapse of a particular kind of patients, and it's difficult to have such enough data for this patient. We had the same problem, for instance, for sarcoma. So you have maybe some 50 or 60 patients. It's not completely sufficient to build a fully supervised classification algorithm with accurate results. So you are always on the edge between enough patients and not enough. So it is why we do proof of concept, and then you have to work with different centers to build some kind of national initiative to be able to build something more convincing from a clinical point of view. But I agree. Yeah, this is not sufficient. This is why I said this is a preliminary.
Okay, thank you. And you have another question. How simple the mechanical model should be in general for clinical purpose? Do you have any comment? Better to have 2 parameters or less for some number?
I think it's easier to start from simple model than complexify. And so 2 parameters. So it's 2 parameters plus initial condition. I think our work more or less shows that it can be useful to have such models. Obviously, there are many things you are neglecting, and in some cases you cannot it's not reasonable to neglect so much. So it really depends on the application you have. But I think we should keep in mind that in current clinical practice, they are not using any serious model besides maybe some very simple extrapolation. For that, sometimes, most of the time, it's not even on the tumor volume, but on the large diameter of the tumor. So we start from very far. So even simple model from our point of view may not be so simple from a clinical point of view.
Okay, thank you.
Okay, so if there are no other questions, we can thank again Olivier. Thank you.
Thanks.